And this is it. This is the yellow thing is the H1. H1 histone. 5 type of histones are there. These histones proteins plus DNA is called. This is the nucleosome structure, and nucleosome structures again called supercoiled and make this chromosome. Eukaryotic chromosome structure. So see this way they packaged. After that, the nucleosome structure, the octamer. Histone core contains 2H2A, 2H2B, 2H3 and 2H4. First they made the dimer, then tetramer, then octamer. And this is the H1 histone. So in the core octamer, H2A, H2B, H3 and H4 present, not H1. This is important for MCQ. So we have studied the genetic code. What is genetic code? We have studied the dictionary that a series of dictionary or code of dictionary that corresponds with sequence of nucleotides and sequence of amino acids. We have read the codon, the anticodons. Codons is present on the mRNA, and anticodon is opposite. Complementary by complementary base pairing, it joined with tRNA. Anticodon is present on tRNA. We have read this. 20 amino acids are there, and for 20 amino acids there are 61 codons. 64 all total, but among them 3 are nonsense codons. We have read this. So what is 61 codons are there, and the 3 are nonsense or stop codon. The characteristics of genetic code we have studied on your study class. If you have any difficulty, you can ask. If anyone has any difficulty, the degeneracy of genetic code in Obel hypothesis. The Obel hypothesis explains why the codes are degenerate. So today we'll start from here. The flow of genetic flow, the flow of genetic information, it occurs from DNA to RNA, RNA to protein. So first happening this, DNA should be duplicated or replicated. Have you been a video of DNA replication? I think you have all seen. So let's start the DNA replication or DNA duplication. The flow of genetic information, let's see what is saying. The flow of genetic information from an individual cell to next generation requires duplication of its genetic blueprint, the genome. DNA replication is required. Replication is the process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. DNA is double-stranded. The DNA structure, it is double-stranded. It is a DNA structure double-stranded. So from this double-stranded DNA, two identical double-stranded DNA should be replicated. It is the most basic process that occurs within a cell. In every cell, it occurs. In prokaryote cell, in eukaryotic cell, the mechanism is little different, but in both cells, DNA replication occurs. In the S phase of interphase, we have studied in school time, 9-10, class 9-10, that in the S phase of interphase, DNA replication occurs. Each time a cell divides, two resulting daughter cells must contain the exactly same genetic information. Exactly same genetic information should be contained on the newly synthesized two daughter cells. In the mitosis, the chromosome number is conserved within each species. We have 46 number of chromosomes. So by this, DNA or chromosome number is conserved. New DNA is synthesized on the orientation 5' to 3'. This is prime 5' to 3' direction. Only in 5' to 3' direction DNA synthesis occurs or DNA replication. So let's see how it is done. So this is, you can see, this is the main or template DNA strand. This is called the template DNA strand from which new daughter strands will be synthesized. They are the templates, And by making these strands as a template, new strands synthesized by semi-conservative way, semi-conservative replication. Why semi-conservative? We'll see that. So it has three steps: initiation, elongation, then termination. So let's see. This is the original template strands and how it is replicated to two daughter strands. DNA replication begin at a specific point called initiation point or origin. This is very important for MCQ question. Your semester will be MCQ based, so you don't have to read all these uh, mechanisms যে কার পরে কি হচ্ছে, you don't have to remember, but remember the things what which called যে কোনটাকে কি বলছে, then which enzyme is needed to uncoil, which enzyme is needed to seal. You have to remember that. So DNA replication begins as at a specific point, and that point is called initiation point or origin. So question এরকম ভাবে আসতে পারে যে DNA replication begins at a specific point called, four options. You have to take origin or initiation point. You have to see what is in the options: initiation point or origin. So mainly origin ही बोले. So it is starting from origin. The events of initiation of DNA replication occurs in two steps. First, so-called initiation protein unwinds a short stretch of DNA double helix at the origin. There is a protein or enzyme. It unwinds the two stretch of DNA. It is coiled double helical. It is coiled. So we have to uncoil this DNA, unzip. The unzipping process, I've shared the video. You can see that again. So it is known as helicase. It is an enzyme, also a protein. We all know that enzymes are proteins. So this protein helicase attaches the attaches from in the origin and thereby breaks apart the hydrogen bonds between the bases of DNA strands. Hydrogen bonds are there between the nitrogenous bases A paired with T and C paired with G. These hydrogen bonds are very strong kind of bonds. These have to be broken with the help of helicase. So helicase break these hydrogen bonds and uncoil or unzip the two strands of DNA. So this is the first step of initiation. To unzipping or break the hydrogen bonds between two nitrogen bases. Thereby pulling apart, so what is done? Helicase attaches Helicase attaches to the origin and breaks apart the hydrogen bonds between the bases of DNA strands. Thereby, it pull apart the two strands. It pull apart the two strands away. As the helicase move along the DNA molecule, it continues breaking these hydrogen bond and spreading the two polynucleotide chains, separating. It moves along 5' to 3' direction. Then what happens? After unzipping, an enzyme known as topoisomerase also helps in unwinding of DNA. It also, along with the helicase, it also helps in unwinding. But the main enzyme is helicase. It unwinds. So remember the function of each enzyme. Important for MCQ questions. Then there is another protein called single-stranded binding proteins or SSBP. It keeps DNA in single-stranded position and thus also known as the helix destabilizing protein HDP. Very important. Helicase, it unwinds the two DNA strands by breaking the hydrogen bonds, but SSBP, it has similar type of function, but it doesn't break the hydrogen bond. It keeps the single-stranded remaining the single-stranded position. It helps to not uncoil the DNA. It helps to remain the template DNA strands in single-stranded position. So it is called the single-stranded binding protein or SSBP. Another name of SSBP is HDP or helix destabilizing protein. Helix destabilizing, helix formation, it um, doesn't help to form it. So DNA again can form the helix because of this single-stranded binding protein or helix destabilizing protein is destabilize the helix by keeping the single-stranded position. So DNA is unwind or uncoiled. So it happens in the initiation, the first step of the DNA replication, the initiation. Double bond or the hydrogen bonds are broken, then the single strand remains, the template strand. So these two strands are called the template strands by the help of this, DNA replication further begins. So let's see what's happened. Second step is elongation. As the try to zoom. As the helices separate the strands by breaking the hydrogen bonds. Another enzyme called primase. There is another enzyme called primase attaches to each strand and produce a primer. So primase is another type of enzyme. It attaches to the each end strand of the DNA. There are separated strands. In the first step, the strands are not separated. It is separating by the help of helicates. See the picture? It is separating. The process of separation occurs. Then in the next step, DNA is already separated. The two strands are already separated. Then what happens? Primase attaches to each strand. So two primases is required. Primase attaches to the each strand. Then DNA polymerase 3 and primase help to add nucleotide bases or small stretch of nucleotide bases that they are the RNA bases because that is RNA primer, RNA kind of enzyme. It attaches short stretch of RNA bases or nitrogenous bases, the very short stretch. So from that DNA replication occurs. Then DNA polymerase 3 wraps itself around its strand and it attaches new nucleotide from the pool to free floating nucleotides surrounding the existing strand to build the new strand. DNA polymerase can only add the DNA bases, but the RNA primase that adds only the RNA stretches. You will be there. Suppose it can attach AUCG TC, this small kind of stretch. But that is U. U is present. But in the DNA, not U, but T will be present. Not U Russell, thymine will be present. So who will remove that? I will tell further later. So after that, after adding this prime help of the primase, a short RNA primer is added. So after adding of the addition of the small primer by the help of primase, DNA polymerase enzyme, in case of this particular DNA polymerase 3, it wraps itself around the strand and it makes the DNA DNA bases A, T, C, G, T, C, 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 C. From this 5' to 3', one strand is synthesized, the leading strand. So let's see what happens next. The nucleotides that make up the new strand are paired with partner nucleotides in the template strand because of their molecular structure. By complementary base pairing, they join. A and T nucleotides always pair with one another, and C and G nucleotides always pair with one another. This phenomenon is known as complementary base pairing. Remember that complementary base pairing. A always pair with T, and C always pair with G, and it regulates the production of two complementary strands of DNA. Two complementary strands of DNA is produced from one template strand. From one template, another strand or newly synthesized daughter strand is produced. So these are complementary to each other. Template and new daughter, they are complementary. So this is the leading strand. How leading strand synthesis I have discussed. Then another strand formation is not easy like leading strand. DNA synthesis produces in the proceeds in the 5' to 3' direction on each strand and in the parental DNA forming two replication forks for each strand. Two replication forks are produced for each strand. On the strand with 3' to 5' orientation, the leading strand, the new DNA is synthesized continuously from 5' to 3' direction. On the strand that has 5' to 3' orientation, the lagging strand, another strand is called lagging strand. The DNA is synthesized discontinuously as a section of short Okazaki fragments that are then joined together. Because DNA should be synthesized only in 5' to 3' direction. This is opposite direction. This is in opposite direction. See the DNA structure. So it has an orientation. Always DNA has orientations. 3' to 5' direction. board থাকলে ভালো হতো এঁকে বোঝালে, but in the Jamboard the drawing is very difficult. So I have shared the videos. Wait, I'll show you. In the biomolecules chapter, we have the DNA structure. So see, this is the DNA structure 5' to 3', another 3' to 5', but DNA synthesis occurs only in 5' to 3' direction. If we can এটাকে যদি turn around করা হয়, if you turn around this DNA structure, then 3' will situate here and 5' here. So from 3' to 5', but DNA synthesis occurs from 5' to 3' direction. So new strand will synthesis at this direction, because 3' এর opposite এখানে 5' হবে, so 5' to 3'. Another strand, this 3' will situate here, the template strand, and the 5' will here, and the 5' to 3' from this direction it will synthesize. But this is opposite. The leading strand will synthesis continuously, but the lagging strand can't. But it has a different 3' to 5' atom. So it will synthesize like this. Many RNA primase are required to attach, like in the videos. If you have seen the videos of DNA replication, it will be easy to understand. Many RNA primase required, and it attaches, and the gaps are sealed by DNA polymerase 3, because the only DNA polymerase enzyme can add DNA bases. RNA primase যেটা add করছে, that is the RNA bases. U is present. So let's see. In the Okazaki fragment, the short stretches are called Okazaki fragment. DNA synthesis process. So Okazaki fragments are formed in the lagging strand in DNA that is synthesized discontinuously as a series of short Okazaki fragments that are then joined together. First হচ্ছে fill করছে in the RNA primase. Many RNA primase come and sit in the new template strand and make a short stretch of RNA bases that is filled with A, U, C, G, C, T, very short stretch. Then the remaining gaps are filled by DNA polymerase enzyme. Once a small segment of Okazaki fragments has been formed, the RNA primase removed the RNA primers are removed from the 5' by the action of 5' to 3' exonuclease activity of DNA polymerase 1. So exonuclease is a type of enzyme. It is called a molecular scissor because it cuts the DNA into a strand, small stretch of DNA it cuts. So by the help of the exonuclease activity, the RNA primers are removed, because the RNA primer should be removed. The RNA bases should be removed from the DNA strand, because that is a DNA strand. Finally, the gaps left between the Okazaki fragments are filled with complementary DNTPs by DNA polymerase 1, and the adjacent 5' and 3' end are joined by DNA ligase. The sealing procedure is done by DNA ligase enzyme. So remember this. This is very important. So first, it is removed the RNA after done the DNA Okazaki fragment are joined. Then after the joining procedure done, then it should be removed. The RNA primers are removed by the help of DNA polymerase 1. Different type of DNA polymerase 1. Then the fragments are Okazaki fragments are filled with complementary DNTPs. RNA primer should be removed. That is done by DNTPs. Then the sealed is present. মানে মাঝখানে যে গ্যাপ থাকে, দেখাতেই হবে in the Jamboard. Wait, I'll show. Wait, I'm showing. From here I can see. RNA primase. Suppose this is a DNA strand, lagging strand. So from 5 prime to 3 prime, it will be synthesized from this direction to this direction. Suppose this is 5 prime, this end is 5 prime, and this end is 3 prime of the newly strand DNA, daughter DNA strand. So RNA primase will come and sit here. Enzyme RNA primase will come and sit here and add some RNA bases or RNA nitrogenous bases, that is AUCG, this kind of base it added. So suppose add করেছে RNA bases, set it AUGC AGCU. Suppose this is the RNA base. Then there is a gap. The gap থাকে, then after small gap, and then DNA polymerase synthesis. Suppose this is 5 prime, this is 3 prime. এই দিক থেকে তো লেখা যাবে না, so I'm writing from this direction. Suppose this is 5 prime, this is 3 prime. 5 prime to 3 prime, it will synthesis. So from here, DNA bases are added by DNA polymerase 3, because only DNA polymerase enzyme can add DNA bases. Suppose it added ATT, CGG, TTTT. Again, from this direction only, the RNA primer should add RNA bases. Suppose it again added AGU, GCCU. Then again, DNA polymerase added DNA bases. Okay, this is going on. Then after termination of this process, the lagging strand, so in between DNA bases, RNA bases are there added by RNA primase. So this should be removed. So who removes that? DNA polymerase, another kind of enzyme, DNA polymerase, by the help of not only one enzyme, but the help of DNA polymerase 1, then DNTPs, it is removed in between the Okazaki fragments. And whose are the Okazaki fragments? These are small stretch of DNAs. They are Okazaki fragments. They are very small. They are DNA fragments. The DNA fragments in between RNA primase in the lagging strand is called Okazaki fragments. It is synthesized by DNA polymerase 3 by the help of DNA polymerase 1 and DNTPs. These are removed. The primers. These are removed. These are removed, then the gap is there. So gap should be filled. So gap is there in between the Okazaki fragments. So gap should be filled. Who fills that gap? DNTPs. DNA polymerase and DNTPs. They fill the gap because DNA polymerase can add only the DNA sequence. So in the lagging strand, DNA polymerase 1 fills the gaps between Okazaki fragments. These are Okazaki fragments. So this is filled. Suppose DNA polymerase can add DNA bases. A T C C C C G suppose এই টাইপের দিয়ে ফিল করছে and in here so you can see এখানে একটু প্রবলেম হচ্ছে because of this. Okay, too easy to understand. I'm removing that. Suppose up to this is a RNA primase removed and by the help of DNA polymerase 1 short stretch of DNA bases are added. And also here RNA from the inner place of RNA primase short stretch of DNA sequences or DNA bases are added. The T's that are present with T's, not U, because this should be a DNA strand, newly synthesized DNA strand. So but you can see there is gap, small stretch of gap, not the place of new nucleotide can be added, but small gap is there. This gap is sealed by DNA ligase. So see the function of DNA ligase. After adding the DNTPs, DNA polymerase 1, those gaps are filled. The adjacent 5' to 3' end are joined by DNA ligase. This is also small stretch of 5' to 3'. Suppose this is 5', this is 3', this is 5', this is 3'. These small stretches also have orientation. So these gaps are joined by or sealed by DNA ligase. DNA ligase is like sewing machine, it is sealing. So problematic এখানে drawing করা. Okay. So it is sealed. Who's sealing? DNA ligase. The problem in so these gaps are then sealed. Then a continuous basis will find. So lagging strand synthesis occurs like this. Leading strand কোনো problem নেই, continuous procedure. But in the lagging strand, first numerous number of RNA primase comes and synthesis the RNA RNA bases. It gives the RNA bases short stretch of RNA sequences. It added. Then the gaps are filled by DNA polymerase 3. DNA polymerase 3 fills the gap or synthesis the DNA strands, DNA sequences. Then RNA primer should be removed. The RNA bases are removed by the help of DNA polymerase 1 and DNTPs. Then these gaps are filled by these gaps again synthesized by DNA polymerase 1. RNA primary জায়গায় gap থাকবে. These gaps are filled by DNA polymerase 1. Added the DNA sequences. Then small stretch যেখানে একটা nucleotide cannot be added. Very small. That is sealed by DNA ligase. So পরপর এরকম ভাবে lagging strand synthesize. See the video again. Will understand more easily. মনে থাকবে পরপর কি হচ্ছে. Then termination. Then after this is elongation procedure. Then after elongation, termination should be occurred. The two replication forks eventually, this is the replication fork-like structure formation, replication fork eventually meet at a terminus region of the circular chromosome. Termination of DNA replication is restricted to this region, termination region. একটা region থাকে termination সেখানে আসলেই it terminates and fall off. Then two strands are synthesized. This is lagging strand, continuous strand, and this is lagging strand. এরকম ভাবে পরপর যেরকম ভাবে বললাম এরকম ভাবে synthesis হচ্ছে. So and when it meets the termination region, it falls off. So more detail এ দরকার নেই. Don't have to read all the mechanisms or এরকম আসবে না question will not come like that যে write about DNA replication because MCQ type of questions will come. So 2 marks questions are jonno I will give I'm making I'll give the suggestions. So remember the enzyme. Which enzyme is doing what? কে কি করছে? Helicase, important enzyme, uncoiling or breaking the double stranded. More specifically, it breaking the double stranded the hydrogen bonds or the double stranded DNA. It breaking the double stranded DNA by breaking the hydrogen bonds between the nitrogenous bases. AT series বন্ডগুলোকে কাট করছে who helicase. Then SSBP কি করছে? It remains the single stranded position. Then what is the oricide or origin? Then the function of RNA primase, function of DNA polymerase 3, function of DNA polymerase 1. Remember that DNA polymerase 3 adding the DNA bases in both leading strand and lagging strand. Also lagging strand also. Faster age DNA গুলো যে in between the RNA primase. DNA polymerase 1 কি করছে? When the RNA primers are removed, that is sealed by DNA polymerase 1. That is done by DNA polymerase 1. And the gaps are sealed by DNA polymerase 1 by adding the RNA primer জায়গাতে যেগুলো ছিল after removing that DNA polymerase 1 is adding the DNA bases. Then ligase কি করছে? Sealing the nicks in between the DNA sequences of lagging strand. ওকে, এইগুলো হচ্ছে ফাংশন। সো এরকমই আসবে question. Okay, so today up to this, next day we'll start transcription. We will again share videos for easy to understand. Next class is only transcription, then next translation for আছে. Okay, thank you. If you have any trouble, see the video again and you can ask me to today. এফডি থাক, এর বেশি পারবি না নিতে। আর টাইম হয়ে গেছে। ওকে, বাই।
Thank you, ma'am.
ওয়েলকাম।
Thank you, ma'am.